Chapter 6, Section 6, Transcription and Archaea and Eukaryote. When it comes to transcription, we're going to see a lot of the same proteins, enzymes, or at least homologs, I should say, not the same, homologs, orthologs, paralogs, all of the allogs between prokaryotes, archaea, and eukaryotes. Problem difference is what the naming is going to be. You're going to have RNA polymerase, you're going to have promoters, you're going to have terminators, you're going to have many of the same things working here. Just the initiation factors and things like that, the promoter regions specifically will be different. And as was shown in the last section, the RNA polymerase itself is going to have different components. The most important sequence you're going to see when it comes to promoters and whatnot is what is referred to as the TATA box, TA rich, because remember this is DNA we're still talking about. Well, this TATA box, you're going to have the TATA binding protein, TBB, which is a transcription factor. It binds the TATA box, recruits in other proteins and whatnot, and all those together recruit in RNA polymerase. You're also going to have what's referred to as a B recognition element, which is upstream of our TATA box. This is where transcription factor B is going to bind, TFB. TFB binds the BRE, which is upstream of the TATA box, where the TBP protein is bound. Oh my God. So, TBP binds the TATA box, TFB binds the BRE, and these two together, along with a few others that aren't being mentioned here, recruit in RNA polymerase, which begins transcription. Oh my God. That's in the eukaryote. I'm sorry, not in the eukaryotes. That's in the archaea. Here's the TATA, here's the BRE. Here they're showing them really close together, but they can be a bit spread out. The init, this is the initiation. This is where everything starts. So TFBP binds, TBP binds. Notice the TFBP where it's situated. It's on the BRE to this init, and it positions. These two together position by putting a bend in the single strand of DNA, which allows for the RNA polymerase to come in. Because see here, the DNA has to bend around. TFBP, TBP, the bent DNA, and everything is positioned where it needs to be. So when RNA polymerase begins, it's right there at the first nucleotide. When it comes to termination and archaea, we're really not quite sure. Okay, we know with prokaryotes, it's the stem loops, hits that GC-rich region, causes stem loops, things destabilize. As we're going to talk about when it comes to eukaryotes, there's specific sequence that's going to sit there and trigger RNA polymerase to just start laying down the poly A tail. You know, a few hundred amino A's as the poly A tail. For us, once we start and we finish an archaea, archaea to an extent has some of these intervening sequences. We got to get rid of the introns. So we're going to have splicing. Remember the whole spliceosome, collection of proteins that bends around, puts the exon to exon with the intervening sequence as the loop, puts the exons in close proximity, cuts, glues it together, and then there you have, you've spliced out the intron. So this splicing not only removes the introns and joins the exons, but if done correctly or in certain ways, you can alter which exons are joined to which exons. So instead of exon 1 to 2, 2 to 3, 3 to 4, it could be 1, 3, 4, 1, 2, 4, however you want to do it. In Archaea, the introns are rare. There are some, but not to the degree you're going to find in all the eukaryotes. So here's an example of that spliceosome. You have the proteins of the that are, the spliceosome-associated proteins that are going to bend that RNA around, bring the two exons in close proximity. The loop here is the intron. It's going to cut and join. Boom. The intron is just released where it'll be tagged and degraded, and the nucleotides recycled. For ours, RNA, especially if it's messenger RNA, remember, got to have a 5' cap. In your cells, any RNA strand that does not have a 5' cap will immediately be targeted, because our cells have spent the past few 100 million years, evolving defenses against RNA viruses. So that's what the cap is for. That's what the poly A tail is for. The RNases, the enzymes that go through and chomp, 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 chomp RNA, especially if it's going to be an RNA virus genome, will not bind to a 5' cap, but will go to a 3' tail and will start cutting. Well, that's okay, because there's 100 to 200 A's there that you can go through and chomp away, which gives the ribosome enough time to hop on and move down the gene itself, making the pre-messenger RNA. So here's the primary transcript, what I just referred to as the pre-mRNA. Going to have a cap put on, special guanosine, and then you're going to have a poly A tail. Then you're going to splice out all of those introns, and then now you have a mature messenger RNA, which can then be trafficked out of the nucleus into the cytoplasm to come into contact with a free ribosome, and it's either going to be transcribed there in the cytoplasm, or that ribosome will halt, go to the nearest endoplasmic reticulum, and then become part of the rough ER.